All right, everyone, welcome to Cafe Sci here on the 6th of January, 2025, our first Cafe Sci for the new year. Thank you all for joining us online tonight. It is my pleasure in a moment to introduce our speaker, Carrie McDonough. But first, I want to thank everyone for joining us tonight. We really couldn't do Cafe Sci without all of the support that you give us. And if you are so inclined to make a donation to support Cafe Sci, that's always an option. But if you would really like to help us out, then when you come next month, we'll talk about next month's speaker at the end of tonight's presentation. When you come next month, go ahead and bring a friend with you, or bring two friends with you, or even three friends, and help spread the word with Cafe Sci. And let's really get everyone talking about some of the current science that's being done here in Pittsburgh and our surrounding region. So I would also like to thank PPG for presenting Cafe Sci. Thank you, PPG. And I would like to uh, go ahead and introduce our speaker for tonight, Dr. Carrie McDonough. Carrie is a chemical oceanographer and analytical chemist working at the intersection of environmental analytical chemistry and chemical biology. She received her bachelor's in chemistry from the Massachusetts Institute of Technology in 2008 and her Ph.D. in chemical oceanography from the University of Rhode Island Graduate School of Oceanography in 2017. She then completed a postdoctoral fellowship at Colorado School of Mines, developing high-resolution mass spectrometry methods to identify and measure per and polyfluoroalkyl substances in biological samples. The McDonough Lab at CMU investigates organic pollutants, how they're transported through the environment, where they end up, and their potential impacts on water quality, environmental quality, and human and ecosystem health. They use HRMS, ion mobility spectrometry, and other advanced analytical strategies to expand detection and deepen understanding of impacts and of undiscovered or overlooked pollutants. Carrie's work emphasizes the importance of evaluating bioavailability, bioaccumulation, and the biological transformation of organic contaminants in environmental risk assessment. And without further ado, I would like to turn things over to Carrie. And remember, you can submit your questions at any time using the Q&A box at the bottom of your screen. Carrie, take it away.
All right. Thank you so much for the introduction, Brad. Share my screen here. Does everything look okay?
Looks good.
Okay, just making sure. All right. Well, thanks everyone for coming. As Brad mentioned, I was originally scheduled to do this in November, and then through things that were outside of everyone's control, ended up getting moved. But this time, when they said, "Oh, the snow, we might have to move things around," I said, "Come hell or high water, I'm talking about PFAS tonight." So here I am to talk about PFAS. We're not rescheduling again. So I'm really glad that everyone could make it, and that no one had to drive in nasty weather to do so. So I'm going to talk about fluorinated organics, fluorinated pollutants tonight. Some of the things that make them different and more difficult to analyze and understand than other well-studied pollutants, and how we kind of use different analytical techniques to characterize fingerprints of these contaminants in biological systems. So how do we take what's in a living organism and kind of backtrack to, okay, what was in the environment to cause this kind of accumulation in a living organism? Is kind of a lot of what my lab does. So to get started, I wanted to just introduce the lab really quickly. So for the past 3 years now, I've been in Pittsburgh running a lab at Carnegie Mellon. We do things kind of on the intersection of environmental chemical biology and analytical chemistry. So we do a lot of analytical techniques, a lot of mass spectrometry, which I'll talk about tonight, um, but all kind of focused on the intersection between environmental contamination and living things. So how do these contaminants interact with and accumulate in living organisms, whether that be humans or wildlife. And one of the central questions that we're addressing is really, we've learned a lot about past chemical releases. Clearly, they've been really detrimental and costly to human and ecosystem health. But there could be current releases, current chemical usage going on at this very moment that could also contribute to a future toxic legacy for our kids and our kids' kids. So a lot of what we're trying to do is use these advanced analytical methods to kind of predict or get, a faster idea than we've had in the past, so that we're not so reactionary when these contaminant issues occur, so that we can kind of predict them better, prevent them better. So environmental chemistry generally focuses a lot on kind of the source of contamination. So where does the contaminant come from? Where does it go in the environment? So its fate, how it's transported there. And where my lab gets super interested is kind of this intersection, fate and exposure. So once something has reached some place in the environment, how then are we exposed to it? Is wildlife exposed to it? And how does that translate from an external exposure, so something that you have in maybe your drinking water or in your air, to an internal dose? So what's actually in your blood, in your body, in your tissues that can cause then a biological effect? So we really focus on that exposure dose kind of fate area. And I also do a lot of interdisciplinary collaborations with a lot of awesome toxicologists, epidemiologists, metabolomics experts to get at that effects question as well. So to start off talking about kind of our work, and particularly our work with fluorinated compounds, I wanted to use this kind of analogy that I find very motivating to kind of explain one of the reasons I like analytical chemistry so much. So I like analytical chemistry because it makes invisible things visible. I can take a sample that looks like clean water, and I can find peaks showing that there are compounds in it that were completely invisible. I think that's awesome. I think all of the principles behind it are really cool, and it's really what sparked a lot of my interest in this field. And I think about it kind of as like a flashlight in the dark, in that we can't protect ourselves from things that we can't see, right? So we have to be able to see or detect hazards in order to do anything or protect ourselves from them. So when you're walking in the forest at night, you need a flashlight. Otherwise, you're probably going to trip over a bunch of different stuff like branches and what have you. But as you're doing that, you have to make a decision, right? When you have a flashlight, your flashlight doesn't just make it daytime. You have to point it in a certain direction. So you can point that, forward, backward, wherever is relevant to you at the time. And in a similar way, when we're using analytical instrumentation, no matter how advanced it is, we have to make choices. So we're never going to see everything all at once. We have to choose to see some specific things based on the amenability and the ability of our instrument to isolate certain compounds. So if we're pointing in a certain direction, we might see, these particular structures. A lot of these are legacy, kind of well understood pollutants. But we have to keep in mind that outside of our flashlight range, there's all sorts of other compounds that could be present and that we may have to be concerned with that we might not even know about. So this is something that I've been really interested in, particularly from the viewpoint of thinking about analytical chemistry through history, and how as we get better and better at this, we get bigger and better flashlights, we're going to find more and more things that maybe we should be worried about. So we're finding more and more hazards, and it's raising more and more questions for us. So to give kind of an example of this analytical progress, I'm going to start by talking about a contaminant that a lot of you have probably been familiar with before, DDT. Which won its creator the Nobel Prize in 1948 because it saved so many lives during World War II by protecting from malaria and other diseases. So DDT was really seen as kind of a really a lifesaver chemical. that was created. It started being used for public sale in the United States in 1945 and really started getting kind of just liberally sprayed around to get rid of harmful and pesky insects. So something that was really just directly applied on the environment in a lot of different places. We then found by the early 60s in 1962, when Rachel Carson published Silent Spring, that DDT was actually really harmful. And one of the reasons that this went from kind of source to effect or cause to effect so quickly is that this was a contaminant that was directly applied. So we knew what we were applying, we were pouring it on the environment on purpose. And the effects were pretty direct. So you could see in places where there are a lot of DDT applications, you could see these effects starting to happen to bird populations, soft shell disease, things like that, that could have led to a complete population collapse for a number of different birds. So it was a pretty immediate effect that was easy to pinpoint onto DDT. Because of that, DDT was banned for most uses except for a few essential uses in 1972 by the EPA. So this is kind of an example of, in some cases, the best case scenario in terms of reactionary types of regulation that we can do. We saw something was harmful and pretty much immediately got rid of it. Even so, DDT is a very legacy persistent pollutant, and so it's actually still in the environment today. So we're still paying for it. So moving on from DDT, if we look at some of the methods that were used to analyze DDT, a lot of times back in the 60s, 70s, the kind of traditional method used was gas chromatography with electron capture detection, which basically is it's a really sensitive method for looking for chlorinated things. So this is what a number of scientists were using in the 60s and 70s when they collected this data here. So here we're looking at GCECD data where you can see DDT, you can see one of its metabolites, DDE, and then there's all these other peaks. So this instrument is picking up everything that's fluorinated in this sample. And we have all of these kind of unknown peaks here, which is interesting. So this is kind of an early example of analytical chemists trying to solve a mystery about unknown pollutants. And what they ended up finding, and Soren Jansen has this really great story about this that he published in 1972, which is cited here. I ended up finding out that all these unknown peaks are polychlorinated biphenyls. There are all these different congeners of fluorinated biphenyls. And some of the ways they found this out mostly centered on mass spectrometry, which is what I mostly use and what I'll be talking about mostly today. So this here is some mass spec data that they collected back in the 60s. So different compounds are coming out of their column and into their detector at different times. They're separating different compounds out by their solubility or their volatility in this case. And then they're also measuring the mass of each of these compounds. So each of these peaks represents some different mass that they're interested in. So that mass gives us some interesting information to start doing identifications. And what they found here is that peaks G, H, I, L, and M, so some of these unknown peaks in their samples, were corresponding to these masses, which were then corresponding to the loss of a hydrogen and the gain of a chlorine. So they found that this seemed to be some kind of chlorinated series. So this is how they were able to figure out that this was PCBs, was by looking at these masses and how the masses changed with this series. They were also able to look at the isotope patterns. This is another really valuable thing we have in mass spectrometry. So you can look at kind of the isotope abundance of a molecule. And chlorine, for example, the natural isotopes that are mostly abundant would be 35 and 37. So you get this kind of characteristic jagged kind of pattern where you've got the +2s being more abundant. So that's kind of a initial, very easy hallmark of chlorinated compounds. So this is a really nice example of analytical chemists using the tools that are available to them even back in the 60s to discover other contaminants. And even these PCBs were also mostly out of use, at least in terms of manufacture and usage, within the 1970s. So again, something that was discovered, found to be harmful, and then regulated. So, in this story, the PCB story that was published in 1972, Soren Jensen also notes in a very prescient way, I think, that similar discoveries of the accumulation of other chemicals are quite likely to occur at any time. So, he's showing us the accumulation of these PCBs that we didn't know about and saying there could be several other things that we don't even know about that could be happening that we'll find out about at any time. What's interesting is at the basically the same time, or even starting a little earlier, there was a parallel story that went quite differently. So fluoride started to be added to drinking water in 1945. And it is one of the kind of big victories of public health by the CDC that has happened since the 50s because it's very protective of teeth. So it can be very, very helpful to help from having dental issues. So a number of dentists at the School of Medicine and Dentistry at University of Rochester were interested as this fluoridation became more popular in understanding, okay, well, we know it's good for teeth. It doesn't seem to have any effects on humans, but we want to know where it goes in the body other than the teeth. It was understood that fluoride just is stored in the bones as well as being excreted from humans, but they were wondering if they could see any difference in the blood levels of fluoride in people who had fluoridated water versus people who did not. So they collected blood samples from Rochester, where they had very low levels of fluoride, and from Newburgh, they had higher levels. And they looked at the blood of those residents to see what those levels looks like. And what they found was a maximum fluoride concentration of 0.1 ppm, average levels about 0.01 to 0.04, and they saw a clear elevation in the population that had fluoridated water. So there's a bit more fluoride in the blood of people who had fluoridated water. 10 years later, so that was in 1950. In 1960, Singer and Armstrong at University of Minnesota came along and did a similar study, and they found very different results. So they found that there was a constancy of plasma fluoride content in individuals with water with a wide range of different fluoride contents. So they're saying it doesn't seem to matter what your fluoride level in your water is, your blood level is going to look about the same. And they found average fluoride levels in human blood to be about 0.15 ppm. So that's about 10 times as high as what Smith et al. Had found. So that's a pretty big discrepancy. So then Donald Taves came along in the late 60s, also at University of Rochester, and was puzzling over these different results. The Singer and Armstrong numbers had kind of become the generally accepted average serum fluoride that we thought was probably what's in most human beings. But he was wondering, why is that 10x off from these other measurements? And his overall takeaway was that these results seemed like they were most easily explained by somehow the fluoride measurements of Singer and Armstrong being too high by some relatively large constant error. So what he then found by trying to kind of recapitulate both of their methods and starting to explore these methods with some blood samples was that a lot of the fluoride content depended on whether or not he ashed the samples. Ashing here meaning burning at a really high temperature, so above 300 C. So if he took blood and fractionated it and looked at particularly the protein fraction and the albumin bound fraction, he found that in the burnt fraction, he was getting almost 10x higher levels of fluoride than he was in the not ashed fraction. So in summary, he's measuring some concentration of fluoride in human blood, then he's burning it and finding much more fluoride. So what this seems like it would mean is that there's some kind of reservoir for fluoride in human blood that is released after burning. And what he started to realize was that this was probably organic fluorocompounds. This is probably fluorine bound to carbon. And even as early as the late 70s, Donald Taves and Warren Guy at University of Rochester were finding that based on NMR results, so they're using nuclear magnetic resonance spectroscopy, it looked like there was widespread contamination of human tissue, human fluids, with trace amounts of these organic fluorocarbons, probably derived from commercial products. So this is well before a lot of this research was done. They were already finding this. And they were finding in their NMR results. So NMR kind of gives you an idea of what structures might be present in a sample, what kinds of functional groups might be present, but it can't really tell you which ones belong to which molecule. Assuming that there was one molecule in this blood sample that they looked at, they decided that maybe it was perfluorooctanoate, which was a known PFAS that was being produced at the time. But they were a little bit on the fence about it because not all of the chemical shifts, so the signals that you get from an NMR, were matching up to this compound. So they had this branched signal that didn't seem like it would be present in PFOA. And they also found that the head group looked like maybe it would be a little bit different. So they weren't quite sure that this was the right compound. And that's where we kind of leave this. So we leave it with a big fat question mark in the 70s, and it doesn't really go anywhere for quite a long time. Taves and Guy weren't really able to get any more funding to keep doing this. They both moved on into other things. So it's a very different story, a very different ending than we saw for those other chlorinated compounds like DDT and PCBs. So then in the late 90s and published in 2001, we had some advancements in analytical chemistry. So we started to use liquid chromatography and electrospray ionization. So this is kind of your basic LC-MS/MS, so liquid chromatography mass spectrometry system, which is a lot of what we use in my lab. So you have a liquid chromatography system that separates out different compounds by their solubility in a complex sample and shoots it into a mass spectrometer where you're using electrospray ionization, which was a new innovation. It's a more soft ionization technique, so it doesn't break apart molecules. It's not as highly energetic, leaves a lot of molecules intact. And it also allows you to basically take a liquid sample, spray it out in a fine mist, and then it undergoes desolvation. So basically, you're going from droplets down to single ions that you can analyze. So this allows you to look for more fragile molecules and also soluble molecules, which PFAS are. So PFAS were really difficult to look for. There were some methods that were pretty complex to do it with the kinds of methods that were used for PCBs and DDT. It was a really difficult thing to look for. So until the advent of these kinds of ESI, LC-MS/MS methods, this wasn't really a routine method that you could be identifying these PFASs in samples. So in 2001, Hansen et al. Published this study showing a method using LC-MS/MS where they identified PFOA, so the C8 perfluorooctanoate and perfluorooctane sulfonate in human blood. So they were demonstrating the method and showing that in solvent, they're not really seeing anything. In laboratory rabbit serum, they're not really seeing anything. But then in human serum, they found that basically analyte-free, so less than 1 nanogram per mil, human serum could not be found. So anywhere they looked, no matter what blood from the Red Cross donations, other general population blood that they used, they could not find clean blood. So basically what this means is that by the time we had a good method to really measure these things routinely, they were already in everyone's blood. They were widespread in our blood before we were able to measure them. So I encourage you all, if you're interested in this stuff, which maybe you are if you're here watching this talk, to also check out, if you haven't yet, this ProPublica article by Sharon Lerner about Chris Hansen. So putting a face to the paper I was just talking about and talking about her story working at 3M when she published that paper and how long it took, how much difficulty she had to go through to push to say, no, my analytical results are right. This is in everyone's blood. She eventually had to go and find archived blood samples from back before PFAS was invented. So before people were starting to produce these things and show, look, this blood from before these were manufactured is clean. And that's how she eventually showed that this was legitimate results. But it took a while and it was a struggle for her. And she came through with it. So it's really useful information for us to have, clearly. So the big question then is what is different about these compounds, right? So we have these legacy compounds that I mentioned, like PCBs and DDT. And then we have these compounds like PFASs, which took a heck of a lot longer to kind of get our heads around and figure out analytical chemistry for, figure out that they were in the environment, any of that stuff. So as a chemist, I think if anyone in the audience is a chemist, you could probably pick out a few things about these groups of molecules that are different. One being that these are anionic, so they're deprotonated. They have anions on them, so they're negatively charged. They're extremely stable because of the carbon-fluorine bonds, but these are also pretty stable. Both groups are fairly hydrophobic, so they don't like to be really dissolved in water. But PFASs are different in that they're oleophobic, so they also don't like fat. And of course, we have the difference that these are fully fluorinated and these are aromatic and somewhat fluorinated. So one of the big differences here is actually the degree of polarizability for these molecules. So I would say polarizability is kind of like malleability of the electrons in the molecules. So when you have a lot of aromatic groups and you're not very polar, you have a lot of electrons that can be pulled here and there to make you more or less attractive to another molecule. So if a molecule comes up with a positive charge, it can induce a charge in that nonpolar molecule and make it more attractive. So molecules like this that are kind of highly inducible or malleable are kind of like big molecules that are just, we think of them as like covered in tape. So it's just sticky, it likes everything. So that's kind of what these types of molecules are like. PFASs are very different. PFASs do not polarize easily. And that's because of the carbon fluorine bonds. There's this really special bond that's extremely stable, doesn't like to break down by any natural processes, and also has a really strong dipole. So our fluorine is very electronegative, pulls all the electrons towards it, making the carbon kind of more electron bare, more positive. And because of that, when another molecule comes up and tries to kind of induce any kind of change in that charge, it's really hard to do. So those kinds of non-specific interactions where it would just be like a piece of tape sticking to another molecule, that's not going to happen. Which is why these molecules are such good surfactants. They don't like anything else, so they're not really going to interact as much with other like things like fatty tissue, organic carbon. So now we know kind of what these molecules are, maybe a little bit about why they're so difficult. So now I'm going to move on to telling you kind of where we are now. Um, so now after, Hansen et al., we have these methods that are widely used now, used by the CDC in their NHANES biomonitoring to look at general population levels of these compounds over time. And we found that these PFASs are present in the blood of, they say, more than 97 or more than 99% of human beings. Really, that means 100%, just maybe a few people are below detection limits, is the way that I would interpret that. So they've really become very widespread in human blood. And if we look at kind of these legacy PFASs, some of the original ones that were detected and monitored, like that PFAS and PFOA in the Hansen et al. Paper, those are declining in our blood over time. So in the general population, they are decreasing over time, which is reassuring, but yes, they are still there. Where do they come from? So the way that they kind of ended up in everyone's blood and ended up being so prevalent for exposure is that they're in a lot of different commercial products and they're highly persistent, so they stick around. So they're in Teflon, used in the manufacture of Teflon and other coatings, used in fabric protector, different kinds of food packaging that are grease resistant or water resistant, carpet protectors, stain resistant, things like that, water resistant clothing, so rain gear. And also a major source of them is aqueous film forming foams. So AFFFs are foams used to fight Class B fuel fires. So the PFASs, because their surfactants actually make kind of like a shell around the fire and starve it of oxygen. So they are very effective for those kinds of fires and were used widely in that field as well. Why do we care about these PFASs being everywhere? One of the main reasons is that we know now that they affect human health, as well as the health of different ecosystems. So they've been associated with a number of different human health effects, including reduced birth weight, different metabolic issues, lower immune response. So children who have higher levels of PFASs in their blood have been found to have lower antibody response to vaccines than children with lower levels of PFASs in their body. Things like diabetes, cholesterol levels, and also some of these have been found to be carcinogens. So actually increasing your risk of cancer. Because of this, NASEMS, the National Academies of Science, Engineering, and Medicine, in 2022 came out with recommended levels, so like a clinical guidance based on 7 different PFASs that doctors can use to kind of look at, if you get your levels of PFASs measured in your blood, should anything different be done in terms of your healthcare? And they said under 2 nanograms per mil, you're probably okay with the usual standard of care. 2 to 20, you should start doing things like screening for dyslipidemia. And then up at more than 20 nanograms per mil, thyroid function testing as well as kidney cancer screening are of concern. So clearly, these things are being taken seriously. They're associated with a lot of different health effects. Some of the mechanisms for these health effects are still a big question mark, so that's still a big area of research. But clearly, the animal studies and the epidemiological studies in humans have shown an association, which is highly concerning and makes us want to know more about what's happening with these compounds, how they accumulate in the human body. So, what my group does is we look at PFAS fingerprints in living things. And this is kind of a schematic of how we get our samples. So, we start with some person who wants to give us a sample. You have to get a trained phlebotomist to come help you get that sample. So you're going to draw blood from this person. Then we take that blood, put it in some vessels, and centrifuge it down. So the traditional way of doing blood monitoring is that you centrifuge the blood, separate out the red blood cells from the serum. So then we just get this kind of liquid protein mix, and that's what we keep to do the analysis on. We throw away these red blood cells. From there, we would put this on an instrument that should be a mass spectrometer, but the only icon that my Excel had was a saxophone, so that's what you get. And from there, we generate data. So you can see kind of the kind of chromatogram we would get from this. It tells us something about the composition of PFASs in this sample. So let's look closer at this chromatogram. So here we're looking at kind of what we would get from my mass spec when we looked at it on the other side. We have retention time, so when things elute from the C18 chromatographic column, and that's going to be related to their hydrophobicity. So we go from shorter chain compounds that are more soluble to longer chain compounds that are more hydrophobic. So as we go up in time, more and more compounds are eluting off of this column and hitting our detector. So that allows us some separation of compounds by their solubility. We can also see some kind of indication of their relative abundance based on their counts per second, and we can calibrate for that using analytical standards so we can actually measure the concentration of these compounds. So here we're seeing kind of short chain compounds up to C8. C8 is the highest that we're seeing. We're seeing 2 different C8s, so these are PFOS, so C8 sulfonates. And we have both branched and linear isomers here, which makes this kind of signature pattern where you have the branched eluting first, followed by the linear. So let's look at 2 different chromatograms for 2 different human cohorts to kind of look at why these fingerprints are useful for us. So we can compare different cohorts and learn something about what their sources might be, what kinds of PFASs might be contaminating their blood or accumulating in them based on what they're exposed to. So here we have a first responder, total perfluoroalkyl acids in their blood at 30 nanograms per mil. And we have an avid seafood consumer from a Long Island cohort that I've been working with. Again, levels at about 38 nanograms per mil serum. And you see pretty different composition in some ways. So the first responder has higher levels of branched PFAS. And then we see these super long chain perfluoroalkyl carboxylates in our seafood consumer that we don't see as much in our first responder. So these are indications of different sources among different humans. So different people have different fingerprints depending on what kinds of sources they're exposed to. We see similar kinds of trends when we look at other species. So here we're looking at, again, that same first responder sample compared to a white shark from the North Atlantic. And you can see this is a very different kind of signature. It's also a very different level, so much lower levels in the plasma of a white shark at 1 nanogram per mil. And here we don't have any of these shorter chain, more soluble compounds. So that whole area is pretty much devoid of PFASs. And then we have all of these longer chain compounds, which are often more associated with biomagnification. So seafood diet, eating fish that have already bioaccumulated a lot of contaminants. When you eat those fish, it magnifies the pollutants up in the food web. And so you end up with higher levels of these ultra long chain PFCAs, which actually kind of mirrors what we saw back here to a lesser extent in a seafood consumer. So it kind of makes sense. Sharks are major seafood consumers and so have this same kind of signature where they have a lot of long chain perfluoroalkyl carboxylates in them. So to give you a little bit of an example of how we can learn stuff from these fingerprints, I'm going to give you some more information about the shark study, which we published last year. So this is a study where we were lucky enough to get 37 different shark plasma samples and a subset of muscle samples as well. I had a master's student who worked with me, Jen Marciano, who did this work for her master's thesis. And we also collaborated with Anne McElroy and Lisa Crawford at Stony Brook, who were kind enough to give us these samples. So they were doing a mercury study on sharks, working with folks at OSearch who go out and monitor sharks every year. So when they go out and monitor their sharks, they take blood samples, muscle samples, and then they re-release the sharks with trackers. And we were able to get these samples to also look for PFASs. So this is kind of what the targeted results looks like for those compounds that we can quantitate in the samples, going from the south coast up to the north. And you can see there's kind of a increase in the total levels. So these are total levels of plasma concentrations in shark plasma, but they're all pretty low. They're all below 3 nanograms per mil. And the most abundant compounds are these, again, ultra long chain PFCAs. So moving on from there, we can learn some things, as you saw in the fingerprints, from looking at these targeted compounds, but it's really just kind of the tip of the iceberg of PFASs. So the vast majority of these studies on PFAS focus on well-studied perfluoroalkyl acids like PFOA, PFAS, the other ones in my targeted method here. So a lot of different perfluoroalkyl acids of different chain lengths is what I have. Things that we have analytical standards for that are fairly easy to quantitate. But there are thousands of PFASs. There are thousands of these structures. If you go look at PubChem, they have a PFAS tree where you can pull up all sorts of different structures and see all of these different PFASs that exist. And the question then that arises is, what are we overlooking potentially that could help us with this kind of fingerprinting? Are there other compounds there that would be more specific to specific sources or that might be really important for knowing things about biological effects that we haven't really looked at yet? So looking for anything beyond targeted PFASs in our sharks. I think I'm the only person who calls these unidentified fluorinated organics, but I like it because I think it's funny. So we looked for UFOs. The truth is out there. And we do that using high resolution mass spec or high HRMS. So why is HRMS useful? Basically, with a normal mass spectrometer, you get mass to charge ratios that usually you can translate as the mass of different molecules. And those would be at unit resolution. So you basically get a whole number, which doesn't give you a ton of information. With high resolution mass spectrometry that we have in our laboratory, we can go out to about 4 decimal points. We get a much more specific exact mass here. So that helps us in a couple ways. It helps us in that the isotope signature and the exact mass help us to understand what the molecular formula is. One of the really useful things here is the mass defect. So each atom, each existing atom, is not the sum of its parts. If you add up the weight of all the neutrons and protons and electrons in that atom, it's not going to quite equal that. It's going to be slightly off by a defect value that is specific to that element. So each element has a different mass defect. Fluorine has a negative mass defect. So a molecule with lots of fluorines is going to have an overall negative mass defect, which you can see here. So the mass defect here is negative 0.02, and for hydrocarbon, it's positive. So this is an interesting kind of signature we can use to start parsing out where fluorinated molecules are in our data. So we use some of this data, this HRMS data, to do things like map by retention time versus exact mass. We can find things like series that are separated by 50 Dalton mass difference, which is indicative of perfluorinated chains. Every CF2 is another 50 Daltons. So that's a nice way to highlight different novel fluorinateds. And then we can also look at this mass defect that I mentioned. So because it's kind of signature to different elemental compositions, you get these nice trends where you have a different mass defect for every different subclass, every different structure, and then you go up in your mass by the chain length. So as your CF2s are added, you get higher and higher mass, but your mass defect stays pretty much the same, and all of your mass defects are going to be negative for these fluorinated compounds. So these are two of the strategies we use to kind of take our complex data and start looking for novel PFASs in it. So let's see what we can find in these sharks using these kinds of techniques. So we took our shark data, we used a suspect screening approach. So basically made a list of exact masses of different theoretical and known PFAS structures. And then we screened our data to see if we had any exact mass matches. We then also did a retention time filtering. So we basically said, we have an idea of what PFASs should look like in terms of when they should elute on a mass spectrometer LC system based on their mass. As they get bigger and more hydrophobic, they should elute later. So we get that trend from molecules that we have standards for, and then we apply it to unknowns. So unknowns should follow a similar trend. And doing that, we found 13 tentative novel PFASs in these shark samples with very low confidence levels. So all of our confidence levels were level 4, which basically means we had an unequivocal molecular formula. We were pretty sure the makeup of the molecules, but we had no idea how those things were connected, what kind of structure we were looking at. So for the amount of work we went through and the advanced analytical instrumentation we used, this is a little disappointing. It's especially important for these kinds of samples. You can't go out and get more blood from white sharks very easily. So it really highlights the importance for these really precious, low volume biological samples that we need to get as much information about their identification of these novel compounds in one injection as we possibly can. So here we got a little bit of information. We can see that the composition of these samples seems to change in different areas. So Nova Scotia has more different kinds of tentatively identified structures than we saw further south, but we really can't go much further than there. So we need other ways to kind of get at these difficult low volume trace level samples to do this kind of full characterization. So my lab is working on different ways to improve PFAS detection and identification in complex matrices, and I will try to be pretty quick about touching on two of these different strategies, one being ion mobility spectrometry, and the other is bioaccumulation directed prioritization. So these are methods we use to kind of combine in vivo and in vitro, so different biological models, to better inform biomonitoring for trace PFASs in precious biological samples. So first I'll show you a little bit about ion mobility, which is a new technique we started using when we moved to Carnegie Mellon. So again, back to our high-res mass spec example, let's look at these two compounds, and we can note that hydrogen and fluorine actually aren't that different in size. So the size of these molecules, just as a molecule that would be flying through space, is not that different, even though their masses are quite different. So this fluorinated molecule is more than twice as heavy as this hydrocarbon, even though their sizes are similar. So we can plot those on a size versus mass chart. And then we can note that if we wanted to get a similar mass for our hydrocarbon, we'd have to have a way bigger molecule, right? So we'd need to have way more CH2 groups. So we would plot that one up here. So way bigger size for a similar mass. So what we see here using ion mobility is that we have different trends for hydrocarbons, a lot of things that are endogenous versus fluorinateds. So fluorinated compounds kind of have their own region on the size versus mass trend line there. So a student of mine who recently defended last year, Dr. Smolinski, who's at the EPA now, used this kind of technique to analyze different residential wastewater composites and pull out things that looked like they might be fluorinated. So you can see here, she had 8,000 different features in this data set, which is a lot of compounds to try and identify. Got it down to 4,000 with the traditional techniques that we would usually use, like taking out the things that are in solvent or background samples. And then using the CCS filter, so the size versus mass filter, she was able to get this down to 103 or 30 compounds, which is a really nice way to filter data. This is a 3D representation of that, where now we're using, again, size versus mass, but we're also using retention time, which I had shown before. So solubility, basically, versus mass. So we're using both dimensions for orthogonal separation to pull out things that look like characteristic PFASs versus all this other junk that's in complex environmental and biological samples, which we can now ignore because we know it doesn't look like it's going to be a fluorinated or even another halogenated molecule. So using these kinds of complementary techniques, she was able to identify a number of different compounds in this wastewater. Pretty promising in terms of really filtering down our data. Another interesting finding here was that she found that the average mass defect of her samples was much lower in treated samples, so samples that had been treated by nitrogen removing biofilters versus those that hadn't. And this is a suggestion that probably there's some kind of transformation happening in the treatment process that she was studying that's causing an increased fluorination kind of character. So more fluorinated molecules, more highly fluorinated after transformation than we saw before. So even without exhaustive characterization of every molecule in the sample, we're able to learn something using this average mass defect in these exact masses. So then my second example here is bioaccumulation directed prioritization. So this is another kind of strategy we're using to try and whittle down what we should be looking at so that we're not just going in blind to trace level samples and trying to find things we've never looked for before. So this work, these examples that I'm going to show you are using aqueous film forming foams, which are highly complex mixtures of known and unknown PFASs. They're often not fully characterized. Here's that mass defect plot again. So this is a characterization of an AFFF that we analyzed that was field collected in the late 90s or the late 80s. And these are all the different structures that are shown by these different shapes. So you can see there's a lot of different molecules in here, from things that are perfluorinated, so fully fluorinated, to things that have all these different hydrocarbon groups on them. So lots of different things in here that we were able to find. And we worry about aqueous film-forming foams because of both indirect and direct exposure. So back when they were used a lot in firefighting and military practice, they were used in things like firefighter training, used at training areas really widely and often without PPE, so without protective gear. So here we're really worried about direct exposure to that mixture. And they're also highly relevant in terms of indirect exposure. So farther away from the source of contamination, those fire training areas, AFFs were left to kind of leach into the groundwater, got into the groundwater, and eventually started to be widespread in drinking water of communities throughout the United States and throughout the world. So it's become a really, really concerning source of contamination in drinking water through these indirect sources. So you're getting kind of a more transformed, more aged version of the AFFF in those situations. So for these studies that I'm going to show you an example of, we worked with Jamie DeWitt, who's now at Oregon State, to dose C57 Black 6 mice with a AFFF. So we took the same AFFF actually that was used in these studies. So it's the same field collected mixture. And we dosed mice for 10 days, followed by a 6-day deparation, which means they weren't being dosed anymore. They were just sitting, kind of excreting. And throughout the study, we collected urine. At the end of the study, we collected blood serum. From this, using high resolution mass spectrometry, I was able to plot all of the novel fluorinateds that I saw by retention time versus mass. And the size of the circles here is related to their abundance, so how big of a signal I got for each of these compounds. And you can see there's a number of compounds really kind of sticking out here, like this oxygen substituted, unsaturated, and ketone substituted perfluorooctane sulfonate. So there's some of the structures we saw on the side here. These are all novel substituted PFASs, so perfluoroalkyl sulfonates, most of which there are not analytical standards for. Therefore, there are no toxicity tests on any of these, no regular monitoring, really very little information on these compounds. So then when we look at biomonitoring data from some of these communities that are contaminated indirectly with these aqueous film forming foams, we see some interesting comparisons. So this is a study I did when I was in Colorado, where there were communities south of Peterson Air Force Base that were contaminated with per and polyfluoroalkyl substances. At the time, the health advisory level was 70 nanograms per liter. It's 4 nanograms per liter now, but that triggered some remediation as well as the study that we did there to measure PFASs in the drinking water of this community as well as blood of the residents there. So we found, again, in our targeted data that drinking water near the source zone, so near where the AFFF was thought to have been released, was higher concentrations of PFASs, and then they kind of went lower as we moved away from that source zone. And we also saw this reflected in the blood serum of residents. So if we separated them by water district from the one closest to the source zone where the AFFF release happened to the one farthest away, we did see a significant decline in these levels, which shows that clearly they are accumulating PFASs that are coming from this AFFF contamination source. But the real reason I wanted to show you this data is that we also did this high resolution mass spectrometry screening on these human samples and found that all of the compound classes, the novel classes that we saw, were also ones that were highlighted in the AFFF dosed mice. So these are all compounds that were prioritized in our bioaccumulation directed prioritization study, which is really nice confirmation that what we're doing is helping us to find compounds that are of concern for human exposure. So particularly this unsaturated PFOS was the most highly bioaccumulative in mice dosed with an AFFF and was found in 85% of the 200 people who were monitored in this study at low levels, but still there, which may suggest that it could be something like a source marker. So next thing I kind of wanted to highlight here is precursors. So the other thing we found in the drinking water of this population was perfluoroalkyl sulfonamides. So these are compounds that can transform under environmental and biological conditions to form PFOS and other perfluoroalkyl sulfonates, which we know are toxic. So we also found these in the drinking water throughout these communities, again, in a kind of gradient going from highest to lowest, from near to far for these releases. But then when we looked at serum, so the blood serum of residents again versus the water composition, we saw that these FASs, so these fluoroalkyl sulfonamide precursors, which are these hatched marks here, were only present in the water. We didn't see them in serum at all, which raises the question, do they get into serum? Are they actually exposed to people? Are they transforming in people? Are they just excreted readily? What's happening to these sulfonamides? So if we go back to our mouse model, here's the serum data I showed before. Now let's look at the actual mixture that we used to dose this mouse. So the mixture used to dose the mice had a bunch of sulfonamide precursors in it. So these gray circles here that are no longer present in the mice either. So again, we see a reflection of what we're seeing in the human cohort in the mice. And we have to ask, where are those sulfonamides going? So what do we know about the biological fate of these FASs in humans or in other organisms? So this is kind of what a perfluoroalkyl sulfonamide would look like. These Rs are just different random substituted groups. And they get dealkylated, so the Rs get removed, and eventually we have degradation down to just a sulfonamide, so no more substitutions. Now we have just perfluoroalkyl sulfonamide with two hydrogens. From there are kind of two pathways this can take. It can either be conjugated, which we're seeing here, in which case it becomes more highly soluble and it's more readily excreted from the body, or it can be transformed to a perfluoroalkyl sulfonate like PFOS, which we know is a problematic compound. So we kind of have like a good pathway versus a bad pathway here, and we want to know which ones might be taken after ingestion of a compound like this. So another student, David Dukes, who also recently successfully defended a Ph.D. and went to Emory for a postdoc, did this study looking at different sulfonamide precursors. And we were comparing basically, again, this dosing solution to what accumulated in blood serum and then to what was excreted in urine of these dosed mice. So here we're looking at everything from highly substituted perfluoroalkyl sulfonamides like this compound and many other, these red molecules could be a number of different kinds of propyl groups, propylamines, propanoic acids, all sorts of things. And then we go down again to that kind of lower intermediate product, the FASA or FASA. And we see that in the urine of the mice, we're excreting mostly these FASAs. In the blood of the mice, we're really just seeing this one sulfonoamidoacetic acid and nothing else. Yet in the AFFF that was used in dosing, so what they ingested, we see all these different highly substituted molecules that are no longer present. So David's project was really looking at, where do these FASA precursors go? Are they accumulated? Are they transformed? Or are they excreted? And some of the data we found here using high resolution mass spectrometry showed that we did have some of these conjugates, so these more readily excreted compounds. So we had C4 to C6 FASAs, and we also found in the urine C4 to C6 and glucuronidated FASAs. So more soluble compounds that are more readily excreted. They look something like this. And we're able to recapitulate the formation of those compounds in vitro. So basically incubate the compounds with a liver enzyme cocktail and look at what forms so that we can see, confirm that this is the same molecule that we would see in the mouse. And we saw chain length dependent conjugations. So shorter chain FASAs tend to be more readily conjugated and therefore more readily excreted, whereas something like the C8s, that'd be the FOSA, which is a precursor to PFOS, is barely conjugated at all. It doesn't seem to be a very relevant excretion pathway for that compound. So to summarize that, what we kind of see here, if we look at just the perfluoroalkyl sulfonamides, so these kind of funnel, semi-stable compounds that form from all these different substituted compounds, when we look at what's dosed, We see a number of different FASs, short chains, C2s, no C8s really. Then we look at the serum, we see no FASs at all. And then we can look at things like kidney and liver tissue. And we see that there is accumulation of these FASs in that tissue, especially of fossa, which was not conjugated at all. So here we're seeing kind of a bifurcation of things that are conjugated versus things that are accumulated. The other really important takeaway here is that we have accumulation of FASs in the tissues, so in the kidney and the liver. We have none in the blood serum. One of the reasons this is really important is that most human testing is done using blood serum. FASs don't like blood serum. They actually accumulate in the red blood cell fraction. So much more informative to get a whole blood test of a person rather than a blood serum test. So this basically implies that you could have blood serum clean of these FASs when it's tested, and they could still be accumulating other places in your body, which is definitely of concern. So, to kind of summarize what I went over today, a lot of what we're trying to do is mixture analysis to uncover the exposome in these complex biological precious samples. This kind of biomonitoring involves trace concentrations and limited volumes, often from, things like sharks or from human beings, where you just get, sometimes I only get 100 microliters, maybe half a milliliter of blood that I can work with. So very challenging conditions when you need to do multiple injections for novel compound identification. So we're using in vitro, in vivo, so both, enzymes in dishes, cells in dishes, and also animal exposures, as well as in silico, so computational prediction approaches for this kind of bioaccumulation directed prioritization strategy so that we can look at real mixtures that have both known and unknown molecules and try to identify overlooked relevant compounds that may be bioaccumulative, that may be relevant for exposure studies. And we're using this combination of high resolution mass spectrometry as well as ion mobility spectrometry to kind of open new frontiers for contaminant identification, prioritization, and rapid risk assessment by trying to simplify down these really complex mixtures and pull out things that might be of interest, like fluorinated molecules. So we're using all these techniques to again pull together in vivo models, in vitro models, and use them to inform biomonitoring in wildlife and humans where it's not so easy to look for completely novel molecules. So with that, I'd like to acknowledge all of my collaborators who I've been working with on a number of these different projects, as well as my funding sources and my excellent research group of graduate students, postdoctoral scientists, and undergraduates, as well as my dogs, Millie, who is the lab mascot, who is pictured here, as well as Pickles, who is not pictured because she's too scared of things, but she's still very supportive. And I'm happy to take any questions. Thank you.
Thank you for such a wonderful presentation, Carrie. I'm sure everyone is at home applauding right now. Um, yeah, I will dive into the questions here momentarily. Just a reminder, you can submit your own questions down below in the Q&A box, and I'm going to just go through here chronologically, I think is a good way to start this. So there's a question about the Taz and Guy 1970 study that you mentioned earlier. And this person's wondering if Taz and Guy used H1 or 19F nuclear magnetic resonance on their fluoroorganic analytes.
It was F19. Yeah.
Okay,
So
great.
they were looking at kind of the different fluorinated functional groups that might be in those molecules. Yep.
Great. And we have a question here. Someone is wondering how many fluoroorganic compounds exist in nature or if they're all synthetic.
Yeah, so that's a very good question that has been somewhat debated. Um, the kind of understanding now, the weight of evidence, I think, points to that they really don't form naturally. Like there may be a couple really small, like soluble compounds that are fluorinated. For the most part, fluorinated organics are not natural compounds. Everything from those really long chain molecules that I was showing down to trifluoroacetic acid. All of those appear to be coming from human activity rather than from any kind of natural processes. It seems like that's not a way that they form.
Hmm. Great. And Peggy here says, do you think we will eventually begin seeing detrimental health effects from microplastics similar to what we've historically seen from PFAS? And if that's not your area of expertise, that's okay too. But figured you might have some thoughts about that.
Yeah, it's an interesting question. I think they're both kind of similar in that they're these emerging contaminants that have different toxicokinetics than most of the contaminants we're used to studying. So they go to different places in the body.
Hmm.
Microplastics I've puzzled over for a really long time because I don't the way I think about contaminants entering the body is a free molecule having to get through a membrane. And a microplastic can't be a free molecule getting through a membrane. So I struggle a lot with even understanding microplastics a lot of the time. And I think that it will probably be similar. It is a very similar story in that the analytical techniques are very difficult. They're very different than what we're used to. It's very difficult to get clean background because microplastics are everywhere. There have been some studies where they tried to do things like measure microplastics in placental tissue, and they had to remove all plastic from an operating room, which is almost impossible, or maybe
Right.
is impossible. So microplastics are really challenging, and I think I wouldn't be surprised if they're similar to PFASs in that they have a lot of different effects that are fairly maybe subtle on first glance and, are coming over chronic low level exposure that are really hard to study. So yeah, that's going to be another major frontier.
Thank you. We've got a pair of questions from Yvonne here. The first is there any chemical trend in emerging PFAS, such as newer emerging ones having longer fluoride chains or anything like that?
Yeah, it's a great question. It's actually the opposite. So I didn't get into this too much, but those C8s, so I showed PFAS and PFOA, which are the PFAS has 8 fluorinated carbons, PFOA has 7 fluorinated carbons, and then the carboxylate. Those compounds were banned because they were toxic and found to be bioaccumulative. So there was actually a kind of they phased them out voluntarily in a lot of places, including in the United States. And after that, the trend has been to go towards shorter chains because the thought is they're not as bioaccumulative. They're more soluble. They won't accumulate in the blood and in the tissues as much. That's true. They do have lower bioaccumulation factors. So the overall level is going to be lower on the same exposure. If you drink water with the same level of a C8 and a C4, you're going to have lower levels of the C4 in your body. But they're just as persistent. So none of these compounds can break down from natural processes. So my attitude would be, maybe we shouldn't really be using them for anything but the most essential uses until we understand all of the effects. Because if you keep making these C4s, they don't have anywhere to go. They're just going to build up in the environment. They're not going to be broken down. But yeah, it's going towards shorter chains and also ether linkages. So adding oxygens to kind of try to make them easier to break down is the trends that I've seen.
Okay. And Yvonne's other question is how many of these novel PFAS are trade secrets that we have trouble identifying because we don't have access to their proprietary chemical information versus ones that are formed secondarily in the environment?
Yeah, the trade secrets are a huge part of my work. It's like a
Oh,
huge
wow.
part of what we do and why we have to do it is because we have to reverse engineer things. I have a lot of colleagues who are even doing things. I mean, I've done some of this work where you're actually looking at commercial products. I mean, we do it with the AFFF and also things like cosmetics. Now there's work going on with lithium ion batteries, actually taking the product and trying to analyze what's in it because we don't know, which is a lot of wasted time considering that someone out there who manufactured it probably does know. But it's because it's trade secrets. So I would say the majority of compounds that we study, a lot of them are those kind of trade secret source. associated compounds. Transformation product wise, we have the conjugates and things like that that form, but most PFASs, you're going from different diverse precursors that are source associated to the final products. And the final products are always going to be perfluoroalkyl acids because they're highly persistent. They don't break down anymore.
Mhm.
So you have all these different precursors. They break down and they make PFOS, PFOA, PFHXS. And those compounds are not only not trade secrets or not like unique transformation products, they're really hard to tell where they came from because they're in so many different things. There's been historically released in so many different ways that doing any kind of source apportionment when all you see is the end products after the degradation is in some cases impossible.
Yeah, and this next question, I think, is a bit of a natural extension here that a lot of people might be thinking. Do you have any suggestions for avoiding these chemicals on a daily basis? It sounds from your presentation that this is global, ubiquitous, contamination, but, you know, are there things like Teflon cookware and, throwing away your Gore-Tex jacket, that people might consider?
Yeah, so both of those, unfortunately, are I love Teflon was my favorite for so long. It's so much easier to cook with than like anything else, but we have gotten rid of pretty much all of our Teflon at this point. Reverse osmosis water treatment is kind of a nice, effective, less high maintenance way to treat water at point of use. So treating tap water. Um, yeah, things like Gore-Tex and other textiles. You can find ones that are fairly good, but it takes some doing. Green Science Policy Institute has a website where they actually have a list of different companies that have taken pledges to not have PFASs in their products. You have to be really careful with some of those pledges because they might be defining PFASs as PFOA and PFOS and not the rest of them. So you have to kind of look into it a little bit. Um, but I would look there maybe as a first starting point. But I know that a lot of the Gore-Tex and other hiking gear not only has these compounds and you could be exposed, but also when you're out hiking in pristine places, you could be leaving these compounds behind. So there was actually a study probably a few years ago at this point where they found traces of PFASs on Everest
Oh, wow.
because of people going up there wearing all of this different hiking gear. So we're actually tracking this stuff up to places where no one even lives.
Oh my goodness. Well, buyer beware, I suppose. Um, we've got a question here from Dottie. Was this talk recorded and can we be sent a link to see it again? Dottie, this talk was recorded. We don't customarily send out the link to everybody who registered, but you can find this on the Science Center's YouTube channel in the next couple of days. Just give us a chance to get that uploaded, and you can find that at YouTube. Search for Carnegie Science Center, and it'll be right there in our videos. And then you can also share that with your friends and show them what they're missing here at Cafe Sci, and they'll be more inclined to come back and check out our next presentation. We've got another question here. Are PFAS and or PFOS congeners found in acid mine drainage? And which other organic compounds does acid mine drainage contain?
Hmm. I don't know. I mean, a lot of things like fracking activity have PFAS associated with them just because really any process where you're trying to reduce friction or you need highly stable surfactant technologies, often they're used. But I haven't seen anything on that.
So maybe that is someone we could look for a future cafe side to see if there's some connection with AMD here that someone is studying locally. So thanks for pointing us in that direction, whoever asked that question. We've got Jennifer here asks, do you know uh, how if PFAS gets into cells, is there a receptor or a channel? or are they endocytosed?
So this is a lot of the stuff that I don't look into as much, but I have a lot of close friends who are toxicologists who I trust who are looking into it. Um, there are definitely receptors that they associate with PPAR, CAR, a few others. They're basically things that associate with fatty acids also associate with PFASs because they have very similar structures. So they get carried around by serum albumin. They can go into cells. They can associate with different receptors. But beyond that, the actual yeah, I think they undergo both passive diffusion and active transport into cells probably. There's evidence that oat transporters, so organic anion transporters would make sense, they're organic anions, um, help to get them excreted from the body, but then they also undergo reabsorption, so they have all sorts of different interactions with different receptors that are still being teased out, I guess I would say.
Yeah, it is a very active field of study, that is for sure. Let's see, we've got a question here from Chris. As a former analytical chemist, this was a great talk. So thanks for that input, Chris. We love when people enjoy these talks. Chris graduated when LCMS was just starting to be used. You're doing great work with HRMS now. How does this compare to the old quadrupole GCMS I used to use? So that's the first question, and we'll give you that, and then I'll give you his second question.
I love that question because my PhD was completely done on single quad and triple quad GC-MSs. I didn't move to high res until my postdoc, so I really like those instruments a lot. Um, I think the exact mass kind of example I showed is really where the difference is. Like the fact that you can get that mass defect and get a signature of the elemental composition of a molecule from that exact mass is really powerful. And all the different trends that come along with that of being able to look at exact mass versus mass defect. So it gives us a way to, especially for small molecules in the kind of field that I'm in, to get at least unequivocal molecular formula. And then if we have fragmentation, which we always have a quadrupole in the work that I do, so we're always fragmenting to get more information about structure. Doesn't always work when you have really low level samples. Like those shark samples, we fragmented a bajillion times and they were just too low to get good information from that. So I would say the way it compares is that it's less sensitive usually because it's less targeted. So we're collecting more data. We're not going to see things down at as low of a level. We're not going to get as nice of fragmentation, but we're going to get a much wider screening with that exact mass measurement that gives us the ability to at least tentatively identify things that we don't even have analytical standards for, which we could never do with unit mass resolution.
Great. And the second question that Chris has is, what are some of the cutting edge methods that may show us even greater resolution in the future? So someone out there is working on the next big technical breakthrough. What do you know about that?
So the next big breakthrough, some stuff I've seen recently is using FTICR, so Fourier transform ion cyclotron resonance spectrometry, which is one of those kinds of instrumentation that's still only at really, really big centers where they have like a room that has the mass spec in it. Like you can't have one in your own lab, but they have even higher resolution by far than what we have. And they're starting to, as concerns are growing more and more about PFASs, I've seen some work starting to come out using FTICR to characterize some of these complex mixtures. So that's something that I've been interested in looking into.
Great. We've got two more questions here. So if you are watching and thinking about your question, uh, do open up that Q&A box and let those fingers do the talking for you now. before we wrap up for the night. So Yvonne has another question here. What practical applications do you anticipate this research being used for?
So I think the main application that we are usually thinking about is expanding monitoring lists. So there are all of these kind of standard monitoring protocols put out by, that CDC uses for its NHANES data and for general population screening, that other places use to do biomonitoring, that the EPA puts out validated methods to look for specific molecules. And those methods are usually based on, the best they can do in terms of which compounds seem prevalent and of concern and which compounds have commercially available analytical standards for them to use. But those aren't necessarily all of the compounds that we should be looking for. So the perfluoroalkyl sulfonamides that I showed a lot today, recently, Wellington and a few other companies started selling analytical standards for those so that you can actually measure them better in samples and start making routine monitoring techniques for them. And I would say they're probably like 100 times more important than a lot of the compounds that have been on those lists. They just were overlooked for a long time. So the main thing we're trying to do is kind of help inform the next edition of those lists, what should be added to those lists that should be monitored for more widely, um, which things might help as source markers. So I mentioned the transformation in the end products and how if you're exposed to a lot of precursors, you're going to have these common products. You're not going to be able to tell what the source was. So maybe some of these novel molecules are more indicative of the source. So they could be used for things like source tracing, understanding how people were exposed, where the contamination came from in different environments, things like that. And I think the other practical application that I've found really important is this recommendation of where we should even be looking for them. We've been looking in serum for so long. When I first did that first mouse study, we only collected serum. We didn't even collect red blood cell fraction or whole blood because we didn't even think of it. And now we see that if you don't collect red blood cells, you will not see those perfluoroalkyl sulfonamides. So I think recommendations on how to better monitor and get a better picture of this stuff is one of the main ways.
Yeah. Thank you so much for that. We've got a couple other questions that have come in here too. So someone wants to know, uh, how do you think the regulation of use of PFAS in manufacturing should be handled?
Yeah, that's a good one. You can't always replace fluorinated molecules. They're extremely useful. I know that's true. I think we have to think about ways to either make them more circular, so recycling systems within facilities so that they're not released to the wastewater or to the air. Instead, they're recovered and reused in that facility so it becomes more circular and the emissions go down. Or ways to do, there's a lot of people working on treatment methods. So ways that we can destroy these things before they get into your drinking water or before they get out in the wastewater. They're usually really energy intensive methods where you concentrate the PFASs first and then you destroy them by some high energy technique. Putting things like that in at the end before things are emitted from industry, I think could be a way to get around this. We have to prove mineralization. So we have to prove that we've gone down to fluoride before I'm convinced that we're really doing a good job. But I do think it's possible, and I do think that there are some places where we are probably never going to be able to replace organofluorine.
Yeah, it is quite a handy quite a handy thing to have in our industrial arsenal for sure. Uh, it's hard to put that unring that bell, as it were. And you also touched a little bit here on Kitamama's question, wanting to know about the remediation methods and effectiveness. So, like you said, concentrating down and highly energy intensive processes to destroy that. Is there anything else you wanted to touch on about remediation?
Yeah, I think the concentrate and destroy methods are really promising. There's a ton of people working on it, and I see advances in it every day. So I'm pretty optimistic they'll come up with things. The things to remember there to keep in mind, though, sometimes you see these articles come out that say forever chemicals are no longer forever now that we have this treatment technique, but they're already out in the environment. So unless you're going to filter all of the ocean water and all of the soil in the world, you're not going to get rid of them. I don't mean to be a downer. I just think it's really important to remember that we have done this to the environment. We can't just go and clean it all up. We're not going to reverse all this damage right away. What we can do is turn off the tap and then use these kinds of destructive technologies to clean our drinking water
Mhm.
and to hopefully remove anything from industrial processes that need to keep using them.
Yeah. And we have one last one here from Chris, who you responded to before. Loved those great answers. He knows the state of New Hampshire is very concerned with PFS contamination around the former Pease Air Force Base. They're working on destruction methods. That's very hard. Has your research had any overlap with the research for cleaning up PFAS contamination?
Yeah, so I've co-authored on a couple papers about treatment. It's hard not to because it's such a high interest area. I often joke that I am more focused on the problems than the solutions, which maybe isn't a great way to be, but it's just kind of the way it's worked out. But because we have these advanced analytical techniques, we are very useful in kind of looking at these different treatment techniques and seeing how useful they are. So I have a bunch of different collaborators who are developing different techniques, and then we'll look at, what byproducts are you making? Are you actually fully getting down to fluoride? Or are you making other funky PFASs? A lot of these techniques at first, when they started to do degradative techniques for PFAS and PFOA, there were a lot of papers that came out that were like, look, we're successfully doing this treatment and you can see PFAS disappear, but they never said what they were making. They never said what the byproducts were. That has gotten better. People are showing that now, but a lot of these treatment techniques just make shorter chains. So maybe you can degrade a C8, but you end up with like, once you get to about C4, it's really, really hard to degrade. So you end up with a lot of C4, maybe C3 compounds, which are still persistent and are actually more highly soluble. So then sorptive technologies aren't going to pick them up anymore. So they're actually harder to remove in some ways. Um, so yeah, we've been doing a lot of work with some folks at New Jersey Institute of Technology and some folks at Carnegie Mellon, looking at kind of what do you make from these different destructive techniques.
Yeah. And we have one more that came in here from Jennifer, wondering if bacteria take up PFAS. And I think that just raises the bigger question of bioremediation. Is there anyone working on genetically modifying, bacteria or fungi or any other kind of methods to remediate PFAS?
It's definitely been a ton of work looking into bacterial degradation. I think I saw a fungal paper recently, but I haven't seen that many of those. There were some really interesting papers from Frank Loeffler a year or two ago on degradation of some PFASs by bacterial strains and then incorporation of the fluorine into the cellular membrane. So they're actually using the fluorine, which is wild. There's been a couple of reports of specific strains that may be able to actually degrade these perfluoroalkyl acids, but very few. So I haven't seen that really panning out yet.
Sure. All right. Well, Carrie, thank you so much for your time and for joining us here for Cafe Sci. Your contact information is on the screen there. So if anyone has a question that comes up, should they feel free to email you?
Sure, yeah.
Perfect. I want to thank everyone for joining us here this evening. Make sure you come back on February 3rd. Our speaker will be Dr. Jennifer Sheridan from Carnegie Museum of Natural History. She's the curator of reptiles and amphibians, and she does really awesome field work with frogs in South America. And then March's Cafe Sci will actually be bumped up a week to accommodate a speaker schedule. So that will be coming in the last week of February, not the actual first Monday in March. So watch for that update to hit your emails. Thank you again, Carrie. Thank you for everyone for joining us, and we will see you all next time for Cafe Sci.